as we were discussing various omic technologies, one of the two primary omic technologies that have the most abundant data sets in biological domain are genomics and transcriptomics. So what we will do in the next few classes is to cover some of the basics of genomics, how it is generated, and what are the applications of genomic data in terms of diseases, human diseases. There are various other uses of genomic data, but we'll cover human diseases primarily in this course. However, this is not an exhaustive description of genomics. It's just to give you a sampling of what genomics is used for and transcriptomic is used for, and so that when later on classes come, you are aware of why certain methods and algorithms were developed and how they utilized in terms of application of these algorithms onto analysis of genomics data. So just to recap, the deoxyribonucleic acid, which is a genetic material, which is the hereditary material, consists of 4 letters: adenine, thymine, guanine, and cytosine, right? And DNA is in a double helix form, which has a sugar phosphate backbone, and the two strands of DNA are complementary to each other, so that once you know one sequence of one of the strands of DNA, you can figure out the sequence of the second strand of DNA, and they run anti-parallel directions. So one strand runs top to bottom 5 prime 3 prime, the other one runs bottom to top 5 prime 3 prime. And this is the, as we discussed, is a unique property of DNA throughout the different kinds of life form, including bacteria, viruses, and humans. Another thing that we discussed was that DNA replication is done by DNA polymerase, which is an enzyme which replicates DNA. It always moves in 5 prime to 3 prime direction, right? 5 prime to 3 prime direction. It always has a requirement of 3 prime hydroxyl group to initiate. Therefore, can't do de novo synthesis. Okay. So these are the primary properties of DNA polymerase, which will come into sort of utility in the next few slides. So without spending again too much time on double, like the how is DNA replicated and all, what we will see is how do we generate, how do we read DNA, right? From an unknown organism, if I extract DNA, how do we read it? And that was the biggest challenge. And now the technologies exist that we can read DNA very rapidly, but the basic principle remains the same, as we'll discuss in next few slides. Just to give you an idea of how the chromosome or DNA exists in a cell, so if I were to extract DNA from a human cell, I'll be extracting these structures essentially, right? These structures are called chromosomes, and humans have 22 pairs of what are called autosomes and 1 pair of sex chromosome, which means from 1 to 22, each of the pair of chromosome is identical. One you get from your father, the other one you get from your mother. So they are sort of paired in that sense. So for each copy on the father chromosome, you have a sort of a copy on the maternal chromosome. The final pair of chromosome, which is called sex chromosome, XX is for females, XY is for males. So that's, in females they are still identical, one from father, one from mother, but in males, X chromosome comes from mother and Y chromosome comes from father. Okay. Apart from that, all other chromosomes are pretty much identical. There are differences between the two paternal and maternal chromosomes, but those are sort of those are there. We will not be worried about that at this moment. But they are practically identical in their sequence. So therefore, they are called pairs of chromosomes. Now, different organisms have different quantities of DNA. For example, Saccharomyces cerevisiae is a simple budding yeast which we use. I am sure you would have seen yeast being used for baking cakes and breads. It's also used for brewing purposes. So Saccharomyces cerevisiae genome is about 12 megabases, which means it has 12 million ACGT letters in its whole genome. Human genome is 3 gigabases. That means it has 3 into 10 to the power 9 letters in its genome. So there is different organisms have different sizes of genome, and as the complexity of the organism increases, the size of the genome increases. But that's not always true, because there are certain plants and amphibians and even species of unicellular amoeba that have higher amounts of DNA in their nucleus. So it's not always true, but it generally is true that as the organism complexity increases, the size of the genome also increases, and therefore each of the organism would require different time frames to sequence the whole genome, and we'll come to that in a bit. So in essentially eukaryotic DNA, if you think of a bacterial DNA, so this is, let's say, a bacterial cell. The DNA is usually a circular DNA, and this is smaller in size. So bacterial DNA could be about 3000 into 10 is per 3 bases only. So small amounts of DNA. So 3000 kb in one sense, kilobases. So if you think of this as yeast, this is human, and this is bacteria. So different organisms have different sizes, and in bacteria there is only one chromosome. There is no sort of requirement to sequence multiple chromosomes. In eukaryotes, you will have different chromosomes. So yeast has 16 chromosomes, humans have 22 plus 1 pairs of chromosomes. I'm talking about not pairs but chromosomes, unique chromosomes, such as like another organism called fruit fly, which is commonly used in many molecular biology labs, has 4 chromosomes. So different organisms have different chromosomes. The chromosome length varies, so that there's quite a lot of diversity in the amount of DNA across organisms. So what is genomics? And as we have discussed, and sort of this is essentially the definition of genomics. It's a large scale study of all the DNA that make up an organism or a system, right? So genome is the whole component of DNA. So if this is your cell, eukaryotic cell, this is the nucleus. All of the DNA in this nucleus will be the genome of that organism from where this cell has come. So if it is a human cell, then it will be human genome. If it is a mouse cell, it would be mouse genome. If it's a plant cell, it would be a plant genome, right? So that's essentially, that's the whole component. And genomics in one sense differs significantly from classical genetics, which people have been studying for almost over 100 years, where if you think about central dogma talks about one gene is converted into RNA, that is DNA, which has a gene, it converted into RNA, that is translated into a protein. So it's sort of one gene at a time, and classical genetics has been very useful in understanding the function of single genes, one gene at a time. And genomics, on the other hand, considers all of the genes that make up the organism. So if I am talking about human genome, I am talking about 3 gigabases of DNA, which is approximately 25 to 30,000 genes. I'm not talking about one gene, but all the genes together. So, genomics allows us to take the whole organism's information and figure out what makes up what are all the genes in an organism, what are all the differences between different individuals of a species, and if we are comparing disease versus normal, what is the difference between the genes of a diseased individual versus genes of a normal individual. So, it gives us a more global picture and a more comprehensive picture of what are the differences between organisms, what are the differences between species, what are the differences between individuals of an organism. So, for example, if you sequence any two human individuals, you will find that they vary at a large number of sites along their whole genome. Even siblings differ. So that is what genomics allows you to do. Majority of the genes will not be different between, let's say, siblings or between any two individual human beings, but whatever differences do exist bring about all the differences that we see physically in these human individuals, in individuals of a population. So, genomics essentially gives you a complete picture of the genetic makeup of an organism or of an individual in a population. So, there are the method of reading DNA is essentially same. It's just through technological innovation we have been able to increase the parallelization and the speed at which it can be done. So, what we will do in next few slides is to see what is the basic principle of sequencing DNA, or what is the basic principle of reading an unknown sequence of DNA. So, if I were to take out DNA from an unknown bacteria or from a human cell, or let's say my DNA or your DNA or SARS-CoV virus, right, sort of coming into news again. So, the whole idea is how do we read, right? So, if you remember DNA, just to recap, DNA polymerase moves 3' to 5' direction and requires a 3' hydroxyl group to add a new nucleotide, right. So, if this is your DNA and it is 3', 5', so if it has added a nucleotide here and this has 3' hydroxyl group, it will be able to add another one which would have 3' hydroxyl group, will be able to add another one which will have 3' hydroxyl group, right. It will keep on doing this as long as 3' hydroxyl group is present. This is because of the fact that we are using deoxyribose sugar as a backbone for DNA. Okay. So, one of the first methods and the most successful methods of reading DNA, any DNA, was developed by Frederick Sanger in 1977. He got a Nobel Prize for that, and therefore, all of the sequencing methods are called Sanger sequencing methods. And essentially, it is a method which is based on the function of a DNA polymerase to synthesize a gene sequence or synthesize a DNA sequence. So, if this is the DNA sequence I want to read, and as I said, DNA is complementary, right. So, which means that if I can read the opposite strand of this DNA, I'll be able to determine this exact sequence. And this is what was used as a method of reading. So, reading by sequencing a novel complementary strand of DNA. So, sequencing by synthesis. The innovation that Sanger did was, instead of using a deoxyribose sugar, he uses dideoxyribose sugar. Which means in this sugar molecule, the 3' hydroxyl group is absent. Instead, there is 3' hydrogen group. Now, what would happen if 3' hydroxyl group is absent? So, let us say if I continue this, next is a nucleotide in which the hydroxyl group is absent. DNA polymerase will not be able to continue beyond this point, and this is exactly what he wanted to do. So, sequencing by synthesis uses dideoxynucleotide triphosphate. N here means nucleotide, which could be A, T, C or G, any one of the four, right. So, he used dideoxynucleotide triphosphate, and I'll walk you through this figure, which would give you a very clear idea of why there is a requirement of doing this and how does this allow us to read the DNA sequence. First thing, we require an initial 3' hydroxyl group. As we saw in DNA replication, we had this primase as a enzyme which gave a primer, right. So, usually we can provide a synthetic primer, or the primer could exist in the construct that is being sequenced. So, we will not go into detail of it. Let us say we provide a primer. Now, there is a 3' hydroxyl group present here. So, what you do with this? So, this is in a solution, in a tube, this whole mixture with the template, primer, DNA polymerase is in a tube, small tube. What you do? You add a large amount of equimolar concentrations of all the 4 nucleotides. So, these are normal deoxynucleotide triphosphates. So, these are all normal deoxynucleotide triphosphate. So, that mixture now you divide into equal parts in 4 different tubes. So, this is tube 1, tube 2, tube 3, tube 4. In tube 1, this is already there, this plus this is already there, and you add a small amount of DDATP, that means dideoxyadenine triphosphate. So, only a small amount. Here you add DDTTP, here you add DDCTP, here you add small amount of DDGTP. So, now you have added these in small amounts. These are still the major input that the DNA polymerase will use to keep on adding the DNA sequence, reading the DNA sequence, but these are smaller amounts so that they do not always get integrated into the sequence, but get integrated at a very low frequency. Now, what would happen? Let us just read through one of the steps. Okay, I will clear this off. Okay, so now here I have DDATP. This is my primer sequence, which was GCAT. Now, if the next sequence that needs to be added, so this is my sequence. So, if you see, this is G, this is C, this is A, T, right. Next to be, so if I were to take the first 4, which is covered by this, the next one should be the complementary to this will be an A. So now if DD and DDATP gets incorporated, the sequence will terminate here. But if one of the normal A gets incorporated, it will continue. Here is the same case, right? Now imagine this reaction is happening randomly in each of the tube. In this case, there is a random addition of DDATP. In this case, there is a random addition of DDTTP. So in this case, what would happen? GCAT, A was normal because here there is no DDATP, but now there is DDTTP, and this can get randomly inserted here or here or here. In this case, A can get randomly inserted anywhere, right? So wherever there is an A, there is a low probability that a DDATP will get inserted in tube number 1. In tube number 2, there is a low probability that wherever T has to be inserted, DDTTP will be inserted. Similarly, in 3, wherever C was to be added randomly, the DDCTP will get inserted, and here would be GTP, DDGTP. So if you see this whole component of these sequences, right, what we are essentially doing is allowing the DNA polymerase to keep on extending till it encounters a DDNTP. And if all the polymerization that is happening in each of the tube is random, we will get random insertions of these DDNTPs, and the sequence will be terminated at that point. So this sequence gets terminated here, this sequence gets terminated here, this sequence gets terminated here. Now if you think about this, if I am reading this this sequence, after the 4th one, after this, the one sequence, so 4 is the length of the primer, I will have a sequence which is 5 bases, I will have sequence which is 6 bases, I will have sequence which is 7, 8, 9, 10 and so on. So this, if I erase this out, this is the 4, this is the 5th one, 5 bases, this is 6 bases, this is 7 bases, right, this is 8 bases, this is 9, right. Similarly other numbers will come. So now when I run this, each of the reaction in a single well of a gel, which allows me to resolve these DNA fragments. So these are essentially DNA fragments which I have made through DNA polymerase at 1 base resolution, which is what this gel allows me to do. I should be then able to read. The smallest fragment will be running the fastest and will be here. This is the smallest fragment and this is the longest fragment. Okay. So the smallest fragment is at the bottom. So first fragment that I read is an A because it was only visible in the lane in which I had added the solution from tube number 1. The second fragment is coming from, this is tube number 1, tube number 2, tube number 3, tube number 4, right. So this is all the A's, this is all the T's, this is all the C's, this is all the G's. So if the smallest is A, followed by T, followed by G, followed by T, followed by C, then by A. So if I keep on reading it this way, I will be able to completely read the whole sequence. Because I already knew the first 4 bases from the primer and I know the rest of the sequence, I can figure out what was the original sequence because it's complementary, right. So the green part is the sequence that I determined from the sequencing reaction that I just did, and the gray is the original sequence. So this is how we determine these or read the sequence. Now this technology can be automated and made parallel, can be parallelized, and this is exactly what was done. So just to automate it, so in this case, in the previous case, what we were doing was we were adding ddNTPs, and usually these are they were radio labeled, right? So you will see this as a dark band on a gel. That was like way back in 1970s, 80s and 90s. This is how it was being done. But once we got different fluorescent markers, so for example, now we can, nowadays in automated sequencing, we have blue fluorescent marker attached to a DDCTP, we have black attached to GTP, we have green attached to ATP, and red attached to TTP. So now what can happen is that if I don't really have to do these reactions separately, right, I can do these reactions in a single tube, mix all these four ddNTPs, which are four different labels, right? These are different labels, along with four dNTPs, and let the reaction run, assuming that all the ddNTPs would be randomly inserted, and they will produce fragments of different lengths at one base resolution. Take that whole solution and load it onto a capillary tube where it allows the smallest fragment to run the fastest and the longest fragment to run the slowest at one base resolution. And then you have a laser light with a detector. So as soon as a laser light is shining on this small fragment, it will determine that this is a green color, and green color means an A was inserted. In this case, it will give me a red color, and that means a T was inserted. Blue color means C was inserted, black color means G was inserted. And that's how I will be able to read. And essentially, I'll get a readout which is given by the peaks define the maximum intensity of the particular color, fluorescent color that is being detected, and that will be the sequence of the gene. And this is normally what we do. And actually Sanger sequencing is very much still used these days, especially for shorter reads, right? For example, you must have normally in many labs we do these short read sequences when we are trying to determine whether we have cloned the gene correctly, right, in a plasmid, or whether we are trying to figure out whether when we did this CRISPR-Cas mutation and the mutation has been inserted at the right site at the right, and the right mutation has been inserted, we do Sanger sequencing. We even for spike protein we do Sanger sequencing. Spike protein in COVID we do Sanger sequencing. The reason is that Sanger sequencing is highly accurate. It has an accuracy of 99.99% and still cost effective for doing short read sequences, and also it gives us the longest length of sequencing to be done. So approximately in a single sequence we can approximately get 500 bases, which is not possible in many modern technologies. So this is very much an alive and still used method of sequencing DNA or reading DNA.